brain and behavior. And like me, she's decided that a really good system to focus on is the proboscis system, the whisker system of rodents such as rats, because here we can really look at the coupling between behavior and neural control under a microscope. We know a lot about this system. We know a lot about the neurobiology of it, and people like Mitra have really been pushing to develop the tools so that we can measure the behavior very accurately. And another focus of Mitra's work is on the morphology of the whiskers and the material properties of the whiskers and how they can influence the computation that the whiskers can perform. So it's really a nice demonstration of this principle that the embodiment of the system really makes a difference to how it can operate. So, Mitra.
Great. Thanks so much for inviting me and for the wonderful opportunity to visit Barcelona and to speak with all of you. So I also liked David's idea of bringing people to the front so it can be more interactive, so please feel free to interrupt with questions and ask where things are confusing. So the focus, of course, in my lab is perception, but I want to start by crediting the people who are really responsible for this work. I have a terrific lab group. The work that I'm going to be telling you about today is primarily the work of the top 4 students, Joe, Blythe, Brian, and Chris, but the other people have also made substantial contributions. So my laboratory is interested primarily in the question of sensory perception, and that is, how does an animal take energy in some form or another, either light or sound, chemical or mechanical deformation, and generate behavior out, right? And only by looking at behavior can we infer that the animal has had a perception, right? We can't directly measure perception, we can only measure behavior, and from that we infer that the animal perceived something. And the question is how this capacity exists in the brain. In particular, we're interested in the question of active sensing. Now, you might not think that many of these senses are active, but if you think a little harder, you'll discover that your eye is really not like a camera. And a camera has a pixel array and the light falls in, and each of the pixels in the back of the CCD array, they respond to the light. But your eye is constantly jittering back and forth, making microsaccades and saccades. It's very actively moving. And if your eye stopped moving, you would go transiently blind. You would be just gray out. For the sense of sound, that is in some ways the most passive sense I can think of, right? You can put on headphones, lie completely still, and you can still hear. The hair cells are vibrating. And of course, in order to localize a sound, we use head movements and body movements, and many animals also use pinna movements. That is, movements of the outer ear, like dogs and cats. You've seen them perk up their ears, right, when they're listening for a sound. For chemical sensing, we sniff. And recent work from Upinder Bala's laboratory in India showed that rats can smell in stereo. So they're comparing the sniffing between the 2 nostrils. And finally, mechanical deformation, which is the sense that my laboratory focuses on, clearly requires activity because something has to press in or you have to press in to the surface in order to deform mechanoreceptors. So there are challenges in the study of the neural basis of perception. There are technical challenges, which is that neural recordings are extremely difficult. There's a large diversity of cell types and responses, and as elegantly pointed out by Joe's work in the lobster, functional circuitry is not always the equivalent of anatomical circuitry. And right now, neuroscientists, I think, are highly limited by the inability to monitor small neurons, circuits of neurons in vivo. So as yet, we can't really go in and monitor how each neuron is responding and their full connectivity with each other. This, I think, is the real limit that we're bumping into in neuroscience. So some traditional limitations then on experimental design is that the studies of sensory systems traditionally deliver passive sensory stimuli, neglecting the role of movement. So for decades, we've had monkeys sitting in chairs passively viewing scenes, right? A lot of auditory experiments are done in the anesthetized animal, and the sense of touch is often done passively, so that the monkey holds its hand still and something comes and pokes on the animal. And neurophysiological studies often don't record brain activity in a context of ethologically relevant behaviors. So the idea here is that in traditional studies, you can control everything and you can require the animal to do what you want. But an alternative approach be, let the animal do what it wants, but quantify the behavioral parameters. And that's the approach that Tony's laboratory and a number of laboratories from speakers we've heard here and my laboratory also take. So let's look at the sense of touch. It requires consideration of sensory-motor integration, and our laboratory takes a neuroethologically consistent approach. So what is touch? So Gibson defined active touch in the following way. He says active touch refers to what is ordinarily called touching. This ought to be distinguished from passive touch or being touched. In one case, the impression on the skin is brought about by the perceiver himself, and in the other case, by some outside agency. So let me give you an example of picking up a glass. I think this should work good. So I'm going to blank out the screen here for a minute just to illustrate this idea. If each of you imagines picking up a glass, you have forces and mechanoreceptor deformation on each of these fingers, right? And it occurs in some spatiotemporal pattern, right? You pick up this glass, the forces occur. You attribute the perceptions that you get through those forces and moments to that glass. You say, oh, this glass is a cylinder. Those are attributes of that glass. You feel it as a single external object. Even if you're moving it around in your hand, you feel it as a single glass. It's the object itself. If someone were to come up to you, and you hold your hand out, and somebody comes up and delivers the same stimuli, you don't feel that as a glass. You would feel that just as punctate sensations on your hands. It's very different active versus passive conditions. So that's really a very dramatic exhibition of how the same mechanical stimuli, just the mechanical stimuli, is not sufficient to tell you what the perception will be. Now, something is going on in the brain to transform those signals into the perception of an object.
And it's related to shaping your hand to feel the glass?
Presumably. Nobody really understands what the differences are between active and passive touch. I can't give you the full answer on that. So the problem, though, is that if mechanoreceptors activation were the complete definition of active touch, then behaviors such as locomotion or throwing a ball would all be considered touch. Right, so suppose I'm walking here. Why is this not considered touch? Certainly my feet are touching the ground. Lots of forces, mechanoreceptors are coming in on my feet. The reason that we don't consider that touch is it's locomotion. The intent of my movement is not to touch the ground. The intent of my movement is to get from point A to point B. 1 minute. So passive touch we can define as the activation of exoreceptors resulting from an external force. But active touch, we need to ascribe something to the intent of the animal. Right? So the activation of exoreceptors and proprioceptors during movements intended to elicit a sensation. So there's not only a physiological variable, but a psychological variable. That is the intent of the movement. So you can imagine using a cloth to grasp a hot pan, in which case we wouldn't really say that you touched the pan. We'd say that you grabbed the pan or you grasped the pan. Or you can imagine rubbing your hand along to judge the texture of that same cloth, and that we might call touch. Yes, question?
Yeah, it's about the locomotion not being touching, but I think there are many examples of locomotion where you actually would need to touch just to find your way. For example, on the rocky terrain, or everyone has walked over pebbles that were kind of hurtful to your feet, you know, and then you find your way by both touching and locomoting.
That's right, that's right. So there are many papers that show actually that you're making incredibly detailed use of sensory data coming from different parts of your feet. And you're judging the forces and pressures at different locations on your feet differentially in order to judge the stability of the ground. Now, would we call that, colloquially, would we call that touch? Maybe if you tap the surface, right, to determine, hey, what kind of surface am I going to be walking on? But if you're having a conversation with a person and you suddenly change terrain, you don't have to think too much about that in order to change your locomotion appropriately to maintain stability, right? That can all happen at an unconscious level. And at least colloquially, and according to Gibson, we would not call that, it's just a definition, we would not technically call that active touch. Right?
So you're just saying like it depends on the level of processing in your brain, the exact location of processing in your brain, because I'm sure that all that goes quite close also in the cortex.
I'm saying there's a problem with our definition of active touch, with traditional active definitions of active touch, and you have to consider really carefully what you mean by active touch.
Are you going to redefine it, or are you happy with it?
Well, I think you need to define active touch with respect to the sensory surface in question. So for example, if I hold my hand out like this, and I scratch my left hand with my right hand, I might be performing active touch with this hand, or passive touch with this hand. But suppose that I'm contracting the muscles in this left hand, and I'm scratching it now. Is this active touch or passive touch? I think there's a lot of ambiguities there. I think what we can define very precisely is whether the muscles in a particularly sensory surface are active, and whether that same sensory surface is receiving mechanoreceptor deformation. Whether that mechanoreceptor deformation is caused by the activation of muscles in that sensory surface, or by activation of muscles in another body part, or by an external agent. Those are things I think we can quantify.
What's the information that is missing in the passive touch? Because
What is
active
that's
touch, you're touching and you feel it, but the action is the same when you perform a passive touch, like with walking. You're still touching.
Walking I would not say is not passive touch. I wouldn't say it's touch. People wouldn't call it touch at all, right? That's not passive. You're active. If you're touching.
What's the information that is missing in your brain when you do
So
passive?
it's some undefined psychological variable. I'm simply pointing out we need to be really careful in these in thinking about these definitions. For example, I think the Gibson definition isn't entirely complete.
But when you're walking, you know you're not paying attention to the tactile information from your feet. But if you stand on something uneven, very suddenly,
Then you do.
that's controlling your locomotion.
So the way that I like to define it is, because of the confound with Gibson, I like to say that you can define active somatosensation. So I would argue that active somatosensation occurs when the muscles in the sensory surface in question are active, period. It's a purely physiological definition. If you want to then define touch, then you have to go to psychology.
Well, I think there are two things that can be said about it. Another thing is, if you anticipate to feel something, I'm not talking about muscles. If I anticipate, then I will pay more attention to a certain group of sensors. That's not in this definition. The other thing is that I would like to hear how you think about open loop versus closed loop, because that sounds to be much closer to what you're talking about. So if it's really, are you going to use feedback or anticipate feedback for your motion, yes or no?
Yep.
It sounds more like such.
So you're getting into issues of expectation and cognition, which I don't deal with at this point in the analysis of the whisker data, where I'm purely focused on the mechanics. So Paul Verscher and Tony actually have some really nice models of cerebellum and the cancellation of sensory expectation based on models from Kurt Bell and Marr dating back decades, etc. And I think certainly there are brain structures that are involved in the cancellation of sensory expectation, but that's above and beyond the type of simple sensory motor ideas that I'll be talking about in the whisker system. Yeah.
One point I'd like to make is one of the properties of central pattern generators in general is that they gate particular reflexes at different phases.
That's right.
In fact, these reflexes reverse depending on where they occur in the step cycle.
That's right. And at the level of the dorsal column nuclei, which is equivalent to the trigeminal nuclei in the rat whisker system, you have extensive sensory motor gating based on whether your muscles are active or not. So the same sensory stimulus can either ascend or be gated out depending on the conditions of motor activation of your own muscles. So I think it's really important to think about what we can define physiologically and mechanically versus what needs to be defined psychologically. So there's the intent of the movement, there's whether the muscles are active, and there's whether mechanoreceptors are deforming, and there's whether those mechanoreceptors are deforming based on the activity of the muscles in that sensory surface. And those are the 4 things I can think of to consider when thinking about active touch.
Would you say the purpose of active touch is to improve perception?
So can you hang on to that question? If I don't answer it in a couple slides, ping me again. So this is how I like to classify movement. I think of the types of movements that animals perform as ranging from a goal of displacement. That is, now we're talking about this psychological continuum. The goal is to displace the animal. That is, locomotion is the extreme of that. The goal is to get from A to B. It's the intent of the movement. Then a second type of movement might be an independent limb movement, right? I can move one arm independent of the rest of my body. Now, it's true that I'm contracting other muscles in order to do that, but this movement is generally independent of other movements. Limb movements can either be non-directed, right? You can stretch, you can wiggle around, or you can specifically direct your movements toward an external object. And that grades into movements that have explicitly a sensory goal. And I've put this region in a gray dotted square because this is the region that Gibson would call active touch. Right? So from a historical perspective, he would call only movements that have the intent to manipulate or explore. That is, I think this answers your question, Nathan, to perceive. He would only call these active touch. And I think that's a little misleading, right? Because as Tony pointed out, and as David pointed out, certainly during locomotion, you may be paying a great deal of attention, and you may be making, even if you're not paying attention, you may be making tremendous use of the somatosensory data that comes in on your feet during locomotion. So why doesn't Gibson call that active touch? It's a good question. I would say that that's a form of active somatosensation, but it's not with the goal of manipulating or exploring, and so Gibson would not identify that as active touch.
So wouldn't that just be the definition based on open loop versus closed loop? So motion focused on improving or changing the sensory capacity, that would be like, if you integrate that, that's closed loop. And if it's open loop, you're just making some motion. If you measure something, that's fine, but you're not going to change the motion.
I'm a little worried about that. So you could think of that as one definition of open loop or closed loop, but I'm a little worried about that definition. And the reason is that I think it's pretty difficult to argue that locomotion isn't closed loop. You're certainly making use of sensory data as you locomote.
Yeah,
so
it's closed loop.
So it's closed loop.
It's not closed loop maybe to optimize the function of the sensor.
That's right. It's not, yes, exactly. It's not perfect.
Well.
Perfect. It's not closed loop to optimize a sensory goal. Instead, it's closed loop to optimize a displacement goal. That is, to displace the animal from one point to the other. So sensory motor activity is tricky. It involves not only mechanics, but also psychology. That's the point here. So I think there's some really interesting analogies to be made with evolution as well, right? So in locomotion, each limb is moved in a regular temporal and spatial relationship to the others, right? So the movement of one limb provides information about subsequent movements of the other. And you can see horses and dogs and deer locomote very elegantly, almost instantly after birth. Then there are independent limb movements, which might require extensive coordination between different muscle groups, but they don't necessarily rely on a constrained spatiotemporal movement pattern, as is the case in locomotion. Then there are some comparative studies across mammals that suggest the extension of the corticospinal tract into more caudal levels of the spinal cord allowed movements of the forelimbs and hands to become independent of locomotor patterns or axial body movements. And that's work that dates back a long time, and I wasn't here for it, but I think John Koss gave a really nice talk in which he may have addressed some of this. Did he talk about that at all?
A little bit, yeah.
So it may be that instead of learning to coordinate the movements of right and left limbs, we always think about learning motor coordination, maybe we really learn to discoordinate them through development. Infants, little tiny babies, are born with a walking reflex. If you take a baby under a month old, and you hold them, you support them, you provide them stability, they can walk. They have a walking reflex. It's essential. It seems to be, at least one theory says, it's a central pattern generator left over from our quadrupedal days, right? So there's a CPG in infants. After, I think, about a month, and I might have that date wrong, maybe it's 2 months, maybe it's a month, I'm not sure of the exact date, they lose this CPG and they have to relearn to walk in the appropriate adult bipedal form. But they have this residual, they seem to have this residual reflex when they're infants. So maybe we're really learning to discoordinate our movements. Now, something that's going to come up later in the talk is this idea of exploratory procedures. How is it that we perform these types of movements that Gibson would qualify as active touch? How do we explore different objects? And the best work on this to date has been done by Lederman and Klatsky. This is a paper from 1987 in which they identified several different, what they called exploratory procedures for how we sense an object. And what you can see is we use different motions, fairly intuitive picture of how we explore, but they were the first to really to write down what we do. So when we grasp an object, the very first thing we do is enclosure, right? So if I want to know whether this is a rectangle or a square box or a cup, I enclose it with my hands like this, right? I shape my hands around it. And they called enclosure a broadly sufficient exploratory procedure. That is, you can get a sense, an intuition for what this object is if with just this single movement, you grasp it once, you can get this idea for what it is. Other of these exploratory procedures, such as lateral motion or contour following, provide more details about the object. Okay, so just think about this for a minute. It makes a lot of intuitive sense. You want to have an immediate impression of what an object is. That is, you want to take a glance at an object, a single glance. You maybe know the word glance from vision. You just glance at something and you instantly have a sense for what it is. But if you want the details about that object, then you have to explore in greater detail, right? You have to contour follow or make lots of saccades. So that's what I just said here, that some exploratory procedures are broadly sufficient. They can provide information about many object properties simultaneously. Others are pretty specific. And generally, when we explore objects, it's composed of two stages. You first perform a broadly sufficient exploratory procedure, and then use the one that's most highly specialized to get this desired dimension. So you can identify objects with a single brief haptic glance. And in the same way that we want we have this idea that humans can tell what an object is very quickly, I would like to develop a theory of rat vibrisal perception that would allow the rat to tell what an object is very rapidly with its whiskers. So that's where we're headed. So a few open questions in the sense of touch. How does the animal localize the object, determine the object's shape? When we actively explore an object, we're not really paying attention to the tap, 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 tap of the flux of sensation on our skin. Right? We're paying attention, oh, this is the glass, this is the cup that I'm holding. So we clearly have to either temporally integrate data across one or more sensors, or we have to spatially integrate it across multiple sensors, or both. How do we do this? How do animals do this? This is the big question of sensory perception. So we use the rat vibrisal array as a model system. And some advantages is that the movements are complex, but there are fewer degrees of freedom than hand movements, and certainly fewer degrees of freedom than the octopus has with its 8 limbs. They have a rich behavioral repertoire, and rats can be trained. And they have a very regular topography with associated somatotopic maps. And we can monitor the complete input and output of this creature. So let me give you some examples of rat whisking behavior. Using their whiskers, rats can determine all of an object's position, orientation, size, shape, texture. Whisking behavior is rhythmic, but it varies between 5 and 25 hertz, so it's adaptable. So Tony's already shown you some nice videos of rat whisking behavior. These are fairly old videos, so low quality cameras. This is rat whisking behavior in real time, and this is rat whisking behavior slowed down. So you can see that there's really some fairly complicated things going on. This is a bird's eye view of a rat finding a reward in a pipette. And what you notice is that the whiskers on the right side of the face, the rat's veering off a little bit here, and then the whiskers on the right side of the face touch the pipette, and then it veers back to orient itself towards that reward. Right? Rats can also do amazing things with their whiskers. So here we're going to see examples of the rat moving its whiskers at different frequencies. So in the top graph, we're tracking the average position of the right and left whisker arrays. Here you'll see that one side goes at twice the frequency of the other, and then they get back in sync. And they can also hold one side completely still while they move the other side. This is an example of single-sided whisking. In this particular example, you'll first see the left side stay completely still while the right side moves, and then you'll see the right side stay completely still while the left side moves. All right, so how do these whiskers move, and how do they sense? So the way that they move is really pretty interesting. There are sling muscles that go around each of the whiskers. This is a picture from Dorfel in 1984. So this is a sling muscle here. It goes around the whisker, which is shown here, going into the follicle. And as that sling muscle pulls back, the skin serves as a fulcrum, and the whisker protracts forward. And inside the follicle, this is a cross-section of the follicle from a paper by Ibarra and Rice. They have all different types of receptors. Okay? The names of the receptors are not important. I'm not going to dwell on them. You could spend your entire career looking only at receptors in the follicle. The point is that all of these mechanoreceptors are sensitive to mechanical deformation. That's the only thing they can sense. They deform, they respond. That's all they can possibly do. Yeah?
Very short question. Why are there different sensors? Just to get an idea of...
Nobody really knows.
Okay.
The other advantage is that there's a regular topography to the vibrissyl array. So I think Tony already showed you that the whiskers are arranged in rows and columns. The rows are given letters A through E, and the columns are given numbers 1 through 6. And then there's a group of whiskers way at the back, which are called the straddler whiskers or the Greek whiskers, and those are given Greek columns names. And the whiskers are shorter towards the front of the animal and longer towards the back of the animal. So neurons are arranged in the brain in topographic maps. So I'll show this for the primate system, and then we'll look at it in the rat whisker system. So if you take an electrode and you put it down into primate cortex, primate sensory cortex, and on the right there, what I've shown is a flattened view of the brain. And then you go poke on the primate's thumb, you'll see that a group of neurons responds. And then you take out the electrode and you move it to an adjacent group of neurons, and you see that the first finger responds. And then the middle finger, the fourth finger, and the fifth finger, and finally the wrist and the forearm, and finally the trunk of the animal, that is the body of the animal. And what I want you to notice is that in this neural map here, the size of the trunk region is smaller than almost smaller than the size of the fingers put together, right? So that the brain, the amount of brain devoted to any given body structure is proportional to the number of sensors on that body structure, at least in somatosensory cortex, in the sensory areas, right? So, sorry, this is gosh, it's a monkey. I don't know which one. I'm sorry. It's not my work. So you've surely seen this picture of the sensory homunculus. This sculpture is from the London Museum, and in this picture, the size of each body part is scaled to represent the number of neurons that respond to this body part. So you can see grossly exaggerated hands and lip regions, which are the particularly sensitive areas in the human. You can also do this for the rabbit, cat, monkey, and human. Can you remind me when my talk ends, please?
Well, we have to finish by 2.
By 2. Okay, so I will not go till 2. All right, I'll try to finish in, say, half an hour. Okay. All right. 45 minutes. Let's see how it goes. So let's look at similarities between somatosensory pathways. So in the hand, you have receptors, and the signals from your hand go to dorsal root ganglion, then to the brainstem nuclei, then to thalamus, and then to cortex. Is this familiar to all of you? Or, okay, so you've seen all this in intro neuro classes, right? So this is the classical ascending limnoscal system. And at every processing stage, there is a somatotopic map. And I just highlighted some of the words there, receptors, ganglion, brainstem, thalamus, cortex, because we're going to see them again when we look at the rat whisker system. So again here, the whiskers on the rat the signals project to the trigeminal ganglion, the trigeminal nuclei, thalamus, and cortex. And I hope will the words light up here? No, they don't. But I hope you see these words in common, right? Ganglion, nucleus, thalamus, cortex, right? It's the same essential pathway that carries the information from the whiskers to what we might think of as the top of the brain, sort of a biased view. But that's the same, a very nice analogy to the hand pathway that carries information in the primate. So the last advantage that I mentioned of the rat vibrissyl array is that we can monitor the complete input and output of the vibrissyl system. And that's what we're going to talk about today. So the outline for the rest of this talk is we're going to look at the mechanics of a single vibrissa. What are the primary mechanical variables sufficient for three-dimensional feature extraction? Then we're going to look at the morphology of the vibrissyl array to see how the shape and the structure of it influence the sensory data that's acquired. We're working towards a full dynamic model of the whisker array. We're not there yet, and so I'm just going to show you a single video, a couple of slides, really brief. I can't go into detail on it because the work isn't done yet. And finally, I'll focus some time on new techniques that we have to quantify behavior. So let's look at the coordinates for three-dimensional feature extraction. So suppose that I want to know where an object is in space. I have to have at least 3 pieces of information about that object, either x, y, z, or r, theta, z, or maybe r and theta and phi, right? So I either have to work in Cartesian, cylindrical, or spherical coordinates, right, to identify where every point on that object is. And just due to the nature of the way these whiskers look, we chose to work in cylindrical coordinates. We don't know that the rat codes things in cylindrical coordinates. This is just a mathematical convenience that we happen to impose on the rat. What I want to point out to you is z is going to be the height, all right? And we think that z might be identified by a labeled line code. So you know the whiskers are arranged in rows and columns. So you can imagine that then the top row is A, right? So if a signal comes in at this height, they can say, oh, the A row hit that object. So the A row is coding for that particular height. The E row is coding for some lower height, right? Theta, there's big controversy about theta. This is part of the discussion that Tony and I had during his talk. Nobody really understands how theta is encoded. I don't understand how theta is encoded. As far as I can tell, nobody else does either, and I'm not going to address it in this particular talk, right? It's a big open question. But what I will talk about is radial distance. That is, how far out along the length of the whisker has the rat touched something? So again, it's important. I start off like this, and I often get questions in the middle of the talk that indicate a confusion between theta and r. Right? Theta is this way, this angular position, and r is out along the length of the whisker. Remember, the whiskers are just like your hair, so there's no sensors along the length. So the question is, how does it know, did it get touched here, or did it get touched here? Where along its length did it get touched? That's r. So this is a graphical depiction of what I just said. Theta is the angular position. Z, we think, is identified by an identity code or a labeled line. But because whiskers have no sensors along their length, how are we going to determine radial distance? So we did some mechanical analyses of how these whiskers might bend. And this is sort of the traditional view of passive displacement of the whisker. So many papers are written based on this idea. You take a little piezoelectric crystal, the blue dot, and you take the whisker and you shake it back and forth. Now, what are some problems with this experiment? Well, one of the problems is that you can see that this whisker is behaving like a completely rigid, inflexible beam, right? Whiskers are like hairs, they bend, right? So there's no bending here, so that's one problem. The second issue is that in this experiment, you're shaking the whisker. You're not letting the rat move its own whiskers. And those will cause very different mechanoreceptor deformations in the follicle. We looked at what actually happens in the active rat as the rat actively whisks into objects. And what we noticed is that the whisker is going to bend, certainly. And depending on where that object is placed, so there the object is very close to the rat's face, but if it's very far away, like as in the second video, it will bend very differently. Okay, now that might have bent more than you expected it to, and that's because your intuition based on that red line there is based on a cylindrical whisker. But in fact, the whiskers are tapered, and so a whisker further out, you'll get even more bending as the rat brushes into that object. Now, some nice work from Miguel Nicolelis' lab in 2001 showed that rats can determine radial distance with only a single whisker on each side of the face. So they trained rats to poke their noses into an aperture, and that's shown up at the top here. And when the rat had to decide whether this aperture was narrow or big. So you can see here this is labeled a variable width aperture. That means that the experimenters could change whether the aperture was big or small. The rat poked its head in. If the aperture was big, it had to go to the right. If the aperture was small, it had to go to the left, say. And by that way, they were able to show that rats could tell how wide the gap was, that is, where on their whiskers they'd contacted that object, based only with one whisker on the single side of their face. So how do rats do this? So Alex and Joe in my lab did some force measurements on a whisker, and what they did is they took a whisker here and they mounted it to a pole, and there are some scales here, so we have some sense of micro measurements, and then they imposed forces at different lengths out along this whisker, and they plotted the results. So here are the force deflection curves for the force imposed at, I think, 6 different radial distances, and what you see is that for a radial distance that is far away, that is close, the slope is much steeper than for a radial distance where it's far away. Okay, so the slope in this force deflection curve is directly related to where the force is imposed out along the length of the whisker. And just using principles of linear elasticity from standard beam mechanics, we can come up with an equation that describes how this distance out along the length of the whisker is related to the rate of moment change at the whisker base. So here in this equation, d is the distance out along the whisker, and c is a stiffness-related constant. It's related to Young's modulus and the moment of inertia, the area moment of inertia of the whisker, so geometric quantity. LBT is the length from the base to the tip of the whisker, m is the torque, or the moment at the base, and theta is angular position. So this should make some good intuitive sense to you. If you move your whisker against an object, and you have to exert a lot of force, a lot of moment to push it past the object, you know the object is close in. But if it's far out, if the object is far out along the distance, along the length of the whisker, you don't have to push so hard in order to achieve that same angular rotation. So that's the basic intuition behind this equation. And we can plot this if you just focus on the black, solid black curve here. What you see is that rate of moment change is monotonically related to the distance out at which the whisker contacted that object. Questions?
About the force, you have
Yeah.
zeros in the diagram. What is the direction of the force?
Good question. So in this experiment, the force was always directed directly down. Okay? But in our modeling, we assume that there's no friction. And in the case of no friction, of course, the whisker slips along the object until the force is directed exactly normal along the object. So, yeah. So we had to compensate by a cosine factor.
What happens if the object is moving? Is there a confound between the
That's a
velocity
great,
of the object?
yes, there should be. In theory, there should be. And there's some work in Karl Svoboda's lab where they had rats whisk into a mouse whisker. Or maybe it was mice whisk into a rat whisker. I forget which. And they showed that the mice were not confounded by the movement of the whisker. You would think theoretically there should be, but they weren't. So they must be using some other cue. We have some ideas about what that is. So our explanation is not sufficient. So we could have tested this idea in the real rat, okay? But I know all of you are interested in biomimetics, and so I put the robot part of the talk first. We thought this behavioral experiment would be very difficult to do in a rat because we would have to look exactly at the detailed curvature at the very base of the whiskers and monitor it very carefully and train the rats, and this seemed that it would take 10 years. So we wanted just a plausibility argument. We wanted something to say, yes, this could work. In the real rat, this could work. And so Joe built a whisker array. It looked like this. And then what we did is we took this array and we brushed it against an object. And we chose a face because we are primates and we like faces. And what I want you to notice here is the tremendous amount of slip as the whisker is brushing up against this face. Okay, and that's something that's obvious in retrospect, that if a whisker encounters a local slope that's perpendicular to its plane of rotation, then our equation is great. It works just fine. But if the object is slanted relative to the plane of rotation of the whisker, then the whisker will slip either up or down on that surface. And so something that I glossed over earlier is that what we actually did is not just to measure the single moment at the whisker base, but we actually put strain gauges on all sides of this follicle, and that allowed us to record the moments in 2 dimensions and accommodate for the lateral slip up and down on the surface slope. And so using this type of artificial follicle on the whisker array, we were able to perform feature extraction. And so here's again our picture of the whisker array sweeping over the face. Each of these blue points is a point extracted from the whisker tapping against the face and pulling out that radial distance. And then we spline those together in MATLAB to reconstruct the face. So I want to be very clear that there's no recognition going on here. There's nothing fancy. It's just simple feature extraction. But it's a plausibility argument that this is a way that the rat could also be achieving the same goal. Now there is a problem here, which is the problem of unknown friction, and that's the question you were raising. Why are we reasonable in assuming that the force is always acting normal to the whisker? We're not, right? It certainly doesn't. There will be an axial component. And the magnitude of this lateral slip will depend not only on the slope of the surface, but also on the frictional properties between the whisker and the object. And what we think might aid in this is that sampling at different orientations is likely to aid in the disambiguation of local slope and friction's contributions to this. So rats certainly sample an object at many different orientations. And you can sort of think of this as a while loop, right? While you're unhappy with your local slope estimate, change your orientation, measure the lateral slip, re-estimate your friction coefficient, and re-estimate your local slope. And using this, you can accommodate for different frictional properties and different local slopes of the object. Now, all of this, let me blank this out for a minute here. All of this was based on very small angle deflections. So for the face, everything here we're just tapping, tap, 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 tap against the object. And that's very consistent with data from Tony's lab that shows that rats just really tap their whiskers very gently against the object. But sometimes we also see rats sweep their whiskers against objects. It's not clear how much this is used during exploration. That has yet to be determined. But, and so this is a case where maybe we take the engineering in a way that the biology doesn't go. We want to know what can we get if we do sweep a whisker along an object surface, regardless of whether the rats actually do this or not. So Frank has this nice slide that I'll show in a few minutes that talks about how biomimetics can be used to understand biology, and it can also be used to help engineering. And so here's a case where we're maybe helping engineering, but we may not be learning as much about the biology because we don't know whether rats do this or not. So we can sweep the object sweep the whisker along different objects. And you know, this is based on a lookup table approach, actually. You know, we're still measuring only rate of moment change. Here we're limited to 2 dimensions. We've now extended it to 3 dimensions, but we're now pulling out the continuous data as the whisker moves along the object instead of just tapping against the object. And we've recently extended this to a full whisker paintbrush. We've simulated this in Open Dynamics Engine, using that as a model of the full dynamics. And one of the interesting results we found is that our success in the ability to determine the full object shape depends on the taper of the whisker. Do you know what I mean by taper? Raise your hand if you know what I mean by taper. Oh, let me explain taper. Taper, so this is a cylinder, right? Taper means that it gradually gets smaller. So it's like a cone, taper. This is a cylinder. So here the diameter is constant, and here the diameter is decreasing. So our success in 2 dimensions, we can use either a cylinder or a taper. But for our success in 3 dimensions, because we want to uniquely determine both R and theta sense contact, relies on this tapered configuration. And rat whiskers are tapered, so that's kind of nice. So this is our whisker paintbrush. This is our first prototype of the whisker paintbrush. And we can put this paintbrush on a microscribe. This microscribe simply gives us the 0 in laboratory frame coordinates. Okay, so suppose I touch an object here, and I touch an object over there. That microscribe tells us where is 0, 0, 0 in the room. Tells us the origin in the room. The whiskers tell us the local location of different points on the object. Local here, local there. Okay, so we need to put those together with the microscribe. So the idea is to develop, something tinier than here, put it on the end here. This is not built yet, right? This is an idea that we have. Put the whiskers on the end here and be able to brush over an object and figure out its features. So we have simulations of this. This is one of our first simulations of a catheter brushing over of a paintbrush brushing over a wall. And we replicated our other experiment. So this is an early simulation, and the light blue dots are the calculated contact points, and the dark blue dots, I think, are the actual contact points. So we know the surface of the wall perfectly. We want to see how well we can replicate that. And in more recent data, more recent video, we're able to replicate, I think this will show here. So here we have a surface, and we're gradually filling it in. Right? All of the red points, the dark red, we're too far behind the screen. The light red is right in the screen. So we're able to reconstruct different surfaces by brushing the paintbrush over them. So our next goal is to build this in hardware. Yeah?
So I see the paintbrush seems to be changing its orientation.
That's right.
So how do you keep track of the orientation of the brush?
There are sort of 2 questions buried there. The first is we just chose a random walk to change the orientation and position of the paintbrush. So that's how we chose it. The second is we just assert a 0, 0, 0 in laboratory frame coordinates. And in practice, the way we anticipate implementing that, so solving 0, 0, 0 in laboratory coordinates is not a very interesting problem for us, right? We just say, look, give us a global origin, and we can solve the contact problem with whiskers, right? Solving 0, 0, 0 in laboratory coordinates, gosh, we should do that with magnets or something like this. Or the way we actually plan to do that is with this microscribe. That's the big advantage it gives us. Essentially put our paintbrush on the end of a robot where we know where the origin is. Right? That's not a problem we're interested in solving. Okay, so
I was much more interested in the orientation itself. In order to do the reconstruction, what kind of tolerance do you have for the the orientation of the brush.
We need to know the orientation. We need to know the 3D position and orientation.
I would think with these flexible hairs, you might have some substantial tolerance. In fact, you wouldn't have to have great precision in knowing the orientation of the brush.
That's a good point. We can accommodate to some degree because the hair itself can change orientation. Yeah. Okay, so this is Frank's slide, and it gets at the point I was trying to drive home earlier. That is, we do experiments with these robots, and sometimes they lead us to new application technologies, right? So that's the idea here. This whisker paintbrush is very different than the rat. The rat, the rat is not, you don't hold a rat and move the rat around like this, right? The rat wouldn't have a fit, right? The rat actively moves its whiskers back and forth like that. And this is what we're not doing that with this whisker paintbrush. Nonetheless, I see it as a bio-inspired approach, and it's leading to some novel applications. Now, how might we use these same ideas to think about natural behavior? And so here we're going back to the neuroscience and thinking about what variables did our hardware array need to extract three-dimensional object features. So the whisker had to be tapered like this, and we also needed the position, and the angular velocity, and the rate of moment change. And what I should have listed up there is also the axial force, the force directed axial along the whisker. And we think the rat should need these as well. And so if we go back to the neuroscience, how should we represent coding in these neurons of the rat trigeminal system? Now, in Tony's talk, you saw that some of the ways that we've characterized the whisker cells before, whisker neurons before, is just by do they respond to whisking, or do they respond to touch, or do they respond to pressure, right? And I think that we can interpret these in terms of these primary mechanical variables, so that we should be able to characterize neurons in the rat trigeminal system in terms of the same variables that we use for our robot. I believe that this should be possible. And so Chris Schroeder has started work on this, and I'll only show a couple of slides because it's very preliminary work, where we have IR flood lamps and 2 orthogonal high-speed cameras. The rat is gently restrained. It's in a body suit, but we don't hold its head, and it's tamed to gently appreciate this so it doesn't freak out when we do this. And we can video either the right or the left side while we're recording from the very primary sensory neurons in the trigeminal ganglion. And this is just a very early video of some of our one camera view of the rat very gently whisking into this post here. You see we get really nice isolated spikes, and I wish that I could tell you more, but we're in the midst of analyzing this data. Everything so far, I've just been in contact with my student Chris over the past couple of weeks, everything so far seems to be coming out that if you look in 3 dimensions, that these neurons really are very sensitive to the bending and the forces, exactly as we predict from our robot experiments. Okay, so let's think a little bit about the morphology of the vibrissyl array. So far, I've been focusing on the mechanics just of a single vibrissa. But the rat has many vibrissa all over its face, and it uses them in parallel. How does this work? How does the shape and the structure of the array affect the sensory data acquired? So we know that whiskers are arranged in a regular array. Rostral whiskers tend to curve forward. Caudal whiskers tend to curve backwards, dorsal upwards, and some whiskers curve more than other whiskers. So let's quantify this. So the idea here is if we knew the spatial arrangement of the whiskers, and we choose a whisking trajectory, we should be able to predict the contact patterns that will be generated as a rat explores an object. Let's imagine that we don't want to work with rats anymore. We just want a model of a rat. We want to put this rat in an environment and predict how will the whiskers hit different objects. So our goal here is to obtain a set of equations that describes the full morphology of the array. You give me a whisker, and I tell you the location of every point on that whisker. And so our data set consisted of whiskers from 6 rats. We scanned in 2 dimensions on a flatbed scanner. And then we scanned the vibrissyl array of 3 of those rats on a 3-dimensional scanner. And we characterize the whiskers array using only 8 parameters. And I'll name those 8 parameters there. The first is the arc length of the whisker. All right, so how long the whisker is in arc length. The second is we found that the whiskers are very accurately represented as parabolas. All right, so we can use simply we can write the expression y equals ax squared. Can you see that over there? And this a coefficient, that a coefficient corresponds to this a coefficient here, describing how curved those whiskers are. Then we need to say where the whiskers are located on the cheek. And we do that by modeling the cheek as an ellipsoid and by locating each base point in theta and phi on that ellipsoid, right, in spherical coordinate system. Then we need angles of emergence. So demonstration would be nice here. So how does the whisker come out of the pad, right? So here I have theta, that is the angle in this direction, and phi, that is the angle in this direction. And then there's psi, which is the projection. It's a sort of a funny projection angle. It's shown here. It's the angle projected into this sagittal plane. And then zeta, I can't really physically demonstrate it, zeta is the whisker's orientation on the rat's face. So if the whiskers were straight, we would not need zeta. Turned like this, it would be irrelevant. But the whiskers have an intrinsic curvature. So we need to specify, are they up, are they down, are they forward? So that's this 4th variable, zeta. Using these 8 variables, we're able to write equations for each of the whiskers. We can say that an arbitrary point W of any whisker, we can calculate that as some function of the row of the whisker, the column of the whisker, and the side. Are we on the right or left side? And we simply take this 2-dimensional whisker. They lie in a plane. The whiskers lie in a plane. We take the 2-dimensional whisker, we rotate it into position, and we translate it to the correct base point. Right? So that's what this equation says. We just take this parabolic whisker, we rotate it, we translate it, we put it on the rat's face. And that allows us to generate a model that's accurate to within 5% error, as confirmed by error analysis, out along the rat's face. And here is a video, and what you're going to see is 2 views of the real rat on the left, and the same views of the model on the right. And we can make the model do the same behavior, put it through the same positions as the real rat. So this is the goal of, again, this is aiming towards being able to predict the full pattern of activity that's coming in to the rat's nervous system. So again, here's a video of the rat moving against a surface. Here the whiskers are colored so you can see them better, get a better impression. You can certainly imagine, suppose we put an object in the way. We could then use our results from single vibrissae mechanics to predict the patterns of forces and moments, or torques, that would come in as the whiskers made contact with that object. Okay, so all of this code is open source and it's freely available on the PLOS Computational Biology website. If you're interested in looking at, say, closed loop feedback patterns, that is, how does an animal go out and get data? How does it change its motor activity based on the sensory data it received? This would be a terrific model to look at the contact patterns and then say, okay, now I'm going to make my rat do this other thing based on the contact patterns it received. For example, one project, I can realize it's late in the course, but for those of you who go on to do this type of work, you've seen rats as they scurry along walls, right? Rats follow walls, they follow tunnels really well. How would the rat use the data coming in on its whiskers to follow a wall? You could think of a very nice closed loop control strategy. how far am I from this particular wall? How do I need to turn? What do I need to tell my legs and body to do in order to follow the contour of this wall around corners? Right? So that's this type of thing we envision doing with this model. Okay, so very briefly I'll tell you about constructing a full dynamic model of the whisker array. So far everything we've talked about has been just morphological modeling and quasi-static modeling, but dynamics are fairly different. Now we can start with the quasi-static model here, as I've been discussing, and what I want you to take home from this picture here is that depending on the curvature, the intrinsic curvature of the whisker, the force will be directed in very different orientations. So clearly the intrinsic curvature of the whisker will have a large effect on the sensory data that the rat receives. And so we are examining the effects of intrinsic whisker curvature using a sensor to look at forces as the whisker collides with different objects. And we can take high-speed video of this, or even just photographs of it if we move very slowly, and look at how the whisker is deforming as it makes contact with the sensor. And then simulate these same experiments using an elastic beam model. And what you see here is this sort of a summary of our results. In each plot here, so let's just focus on this very first one. So on the x-axis, I have axial force. That is the force directed along the whisker length. And on the y-axis, I have transverse force. That is the force directed perpendicular to the whisker. And in the first plot here, the object is placed very close to the whisker base. And that's indicated by s equals 25%. In the next plot, we move the object further out. In the next plot, we move it further out. So the object is located at different locations along the whisker length. And in each of these plots, axial force is on the x-axis and transverse force is on the y-axis. We're going to look as the whisker gradually protracts into that object, how do the forces at the base change? Okay, and that's what's shown in this trajectory here. So blue is the start, where theta is very small, and then it pushes in, push, 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 push. And you can see the force is gradually increasing here. But in each of these two curves here, one is concave forward, and the other is concave backwards. All right? And you can see that when the object is close to the base of the whisker, it doesn't make very much difference. But as you move further out along the whisker, if the object's out here, then it makes a big difference. And if the whisker is oriented concave forward, the forces are much greater. All right? So why do we think the rat has whiskers that are oriented concave forward? In part, we think to maximize that axial force directed along the length of the whisker. And there's work from Carl Peterson's lab in Switzerland showing that the axial forces are really very strongly represented in neurons of the trigeminal ganglion. So you can see also that the whiskers will catch on objects, if they're concave forward. And they'll have a longer duration of contact with the object if they're oriented concave forward than if they're oriented concave back. Right? They get pushed differently.
How much do you think the orientation of the whiskers is reactively controlled?
The only data I have on that is from the Knudsen paper, showing that they change orientation during active whisking. I think the primary way they do it is with head movements.
Yeah, I think that they just say that that's something that happens when the whiskers go forward and they rotate. But I don't have any strong evidence for this, but I think that they may be able to.
Oh, to actively rotate? Gosh, I don't know.
Sometimes you see them concave back, sometimes concave forward.
You sure that's not just a projection thing?
I don't think it's entirely explained by evidence, but I can't.
Interesting. More work is needed, right? Okay, so this is a video my student made looking at full array protraction and looking at how the whiskers change curvature. So the black is concave forward and the gray is concave backwards. And so they change a little bit. This is making use of the Knudsen data. These are just 3 different views of that, how they change a little bit.
So that's not including the rotation.
This does include the roll. It does include the roll. You can see how they're changing a little bit there.
It doesn't seem as pronounced, so it might be.
It's very small effect. Okay, so to get accurate computation of whisker dynamics, we have two approaches. The first is Open Dynamics Engine, and you've already seen some simulations of that with the whisker paintbrush. And the second is we've been working with Todd Murphy and Vlad, a student of his, Vlad Sagete, to work to use a variational integrator approach to model the full dynamics of the whisker array. And this is the data that I said I can't talk much about because it's work in progress and I don't have the full results here. But we're trying to model the collisions of the whisker array as it contacts the wall.
Sorry.
Yeah.
Since the length of the whiskers vary, is that being considered in model as well?
Yes, an excellent question. So the lengths of the whiskers vary a lot. And so, one method that you could think that the rat might use to determine distance to an object is maybe a long whisker hit, but a short whisker didn't. So we know the object has to be somewhere in between those 2 points. We certainly think the rat does make use of that type of information. And, biology uses every piece of information it can get. But maybe your question was asking more in the dynamic simulation, do we take into account the different lengths of the whisker? And the answer is yes. We take into account the different lengths and the different diameters and the material properties of the whisker. We take into account everything we know about the whisker we include in the simulations.
Another question I had was, yes, you just said that the varying length is necessary as it gives more information, but the curvature, the convex and concave, is it necessary to model just the way it is in the rat? And how is that used?
It's a great question. I wish I could give you a good answer. We don't know why the whiskers are curved. They seem to increase forces at the base, and that's an advantage, but we have not shown that it's necessary. We've been able to show that taper is necessary, but the intrinsic curvature is an excellent question. We don't fully understand why. Yeah?
It would change the direction of the contact forces, right?
Yes, it certainly changed. So, yes, the curvature certainly changes the direction of contact forces. So, you can see that when it's curved this way, right, the force has these particular components. When it's curved in this other way, the forces are quite different.
So the bending moments depend very strongly on the curvature?
The bending moment doesn't so much. The force components do. Bending moment doesn't a whole lot.
But it's clearly like if you look at the cross product of radius and force,
yeah,
it's going to be...
it matters some, but not as much as the axial component of the force. The axial component, because the moment, remember, is related to the transverse force. So not as much as the force. As you move out further along the length of the whisker, right? So these are the 2 forces shown for each of the radial distances. And you can see that as you move out further along the length of the whisker, it matters more for the 2 forces.
So the whiskers tend to curve back?
No, forward.
They go forward?
Yes, they tend to curve forward, concave forward.
They tend to curve down, and then when they move, they're rotating.
Let me explain. When I say they curve forward, what I mean is anything in this hemisphere, anything in this hemifield. This would be concave forward, concave forward, concave forward. This would be perfectly concave forward. Concave forward, concave forward, concave forward. You're right. They tend to curve concave forward and down, right? Anything in this back hemifield that I can't do, that would be called concave backwards.
It increases the surface area of the whisker field for relative to the angles at the base.
Maybe. See, we just finished a 3-dimensional scan of seal whiskers, and they're opposite. They're concave backwards. The rat whiskers are concave forwards. They're in water. That's right.
And they're used for different things as well.
They're
sensing flow rather than tactile interception.
Rats may also be sensing flow.
There could be an effect of gravity if they're running out of their droop.
There could be an effect of gravity. I have wanted for a while to do the calculation of weight compared to stiffness. I've been meaning to do that. It's a nice calculation to do.
Also, the most relevant objects are, I guess, more towards the ground. Like if you're doing some following, glass following or something.
Except that they spend a lot of time in tunnels, so then the ceilings are interesting.
Yeah.
But, yeah.
So people, sorry, speak louder. No, go ahead. Wait.
Just commenting that in tunnels, the ceilings are important, and they do a lot of rearing to explore areas above the head of the wrist.
Yeah. Tony's pointing out that if I'm a rat, right, so I'm walking along like this, I also go like this a lot. They rear, they sniff up top, and they feel the top of their tunnels. So rats rear their heads a great deal as well. So, okay, all of this is nice. We have robots, we have some theory, we have models, but what it's really critically going to depend on is behavior, right? And I have a very long neurobiological story about how I think the rats are exploring, and I'm not going to go into it in detail here. I'm going to show you the highlights, I'm going to explain the result, I'm going to show you some nice videos, and I'm delighted to talk with anybody in detail about the neuroscience later, but I thought you probably wanted to hear about the relationship to biomimetics and biorobotics. So we've developed the laser light sheet to monitor the full contact patterns of the whiskers on different objects, and this is work of Blythe Tell. And the idea here is that we have two high-speed video cameras and a collimated sheet of light that goes in front of a glass, and we can monitor the rat's whiskers as they make contact with this sheet. And that's what's shown in this picture here. So we can identify each of the whiskers in this picture. This is very similar to work that Frank is doing. The octopus is looking at the video recordings as the octopus suckers move along an object. So this is a high-speed video of the data that we get from these whiskers. So this is a bird's eye view of a rat, and you notice the rat has an implant on its head. That's because we can also get neural data at the same time. That's what we're pushing towards. So we see the front-on view and the top view of the whiskers. And then we use PTV to track the whiskers. We treat them as particles, and we solve the correspondence problem that way. We identify the whiskers using undergraduates. So we hire undergraduates to come into our lab, and we train them to identify each of the whiskers, and they have to identify the whisker the first time it makes contact, and then the PTV algorithm follows it for the rest of the time. And in the experiment that I'm about to describe, we took data from 5 naive rats, and we recorded during the first few whisks of exploring this flat glass wall, and we looked at over 2,000 whisker contacts. And this is the data we obtained. So we call this painting the object. So really complicated exploratory patterns. I'll play that again just because it's nice. So we've arbitrarily chosen different colors for the different whiskers. Whiskers that are close together are the same colors. Yeah.
There's also, you could have also colored them according to the brain regions. That would have been interesting.
According to the brain regions. Well, the whiskers are the brain regions.
Yeah, but you made this division, right? You showed us initially. Like different rows of this.
Oh, this is a row, right? The yellow is a row, the red is a row, orange is a row, green is a row, blue is a row.
Oh, so it's not that arbitrary.
No.
Can you explain a bit more about the technology? So the laser light sheet comes down and is it very thin?
It's 2 millimeters.
So it's all right, okay.
Mm-hmm. The
reason the resolution for
the reason we had to make it so thick is that the whiskers move so fast that unless we had made it that thick, we wouldn't have been able to see the motion, see them make contact. We can make it very thin, 100 microns thin, but then the whiskers are in and out so quickly we don't see it.
So what's the spatial and temporal resolution you can get?
I would have to look in the paper. I can give you a sense for that here. So this is the video that you just saw. And these are the contact points that we're able to uniquely identify. But I should know that, and I can look in the paper and find it for you.
Sort of roughly?
Sorry, I don't.
Sub-millisecond?
Oh, well, the temporal resolution is limited by our high-speed cameras, and that's 1,000 frames per second.
Right, okay.
The spatial resolution, that's the one I'm less confident of because I don't know the error regions around each of these points. But these points, I know that these points are distinct. So the error regions have to be smaller than the minimum distance between those points.
So the very tip of the whisker is extremely thin.
Yes, it is. We do think we're getting those very tips. And we calibrated we calibrated putting an isolated whisker on a motor, and we tapped at different locations on the surface to ensure uniformity and to ensure that we were getting the very tips of the whiskers. So I'm confident we're getting everything, but I can't tell you the exact error measurements without looking at the paper. That's in our IEEE paper. So again, I said I wouldn't go into the details here. One important point is that the rat tends to fixate at a location and take several samples, and then it moves a lot, and it takes several samples at a new location. And this is, we make the analogy of eye movements. We think of this as tactile saccades. So it puts its whiskers here, whisk, 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 then jump, make a saccade, whisk, whisk, whisk. So we like this analogy to eye movements. And again, you know, I can go through lots of details here, but the idea that we're really driving home towards is that each whisk seems to contain a window of time where these whiskers really are sampling the object, right? So each whisk takes about 125 milliseconds, but there's a very brief window of time. I wonder which, yeah. A very brief window of time. We call this the collective contact interval, when the whiskers are maintaining contact with the object. So let me explain this plot in some detail. So on this axis we have time, and on this axis we're plotting the whiskers. All right, so this is just all the whiskers in order, plotted by row, and within each of the rows we have the different columns. So all the whiskers here. When they're in contact with the object, we paint it black. When they're not in contact with the object, it's white. And when a particular whisker never made contact with the object during the entire whisk, it's gray. So what you see is that the whiskers are sort of scattering onto the object, and that's shown by this gradual rise in the black traces across the array. And then there's this interval here that we define, this collective contact interval, when 80% of the whiskers that are going to be in contact with the object are in contact with the object, and they're maintaining contact with the object. Now, this is the duration of a whisk, from 0 to 125 milliseconds. And it's this really tiny window within this whisk, when we think the rat is really acquiring the sensory data. And that's when they're sustaining contact with the object. Because if we look at the motivation for this is that if we look at the patterns of activity that happen when the whiskers collide with the object, the temporal patterns seem to be unstructured. There seems to be very little information about the object as the whiskers tap onto this object. So we think this information might be present in this sustained interval, right? That's what the data seem to suggest. So this is sort of a cartoon of how we think the data is being acquired, that there's this relationship between the head velocity. So you saw that the head is sampling at one location and then sampling at another location. The head velocity is oscillating. The whisker angle, that is how they're doing the whisk cycle, is also oscillating out of phase with the head velocity. The contacts are occurring as the whiskers protract forward. And then there are these intervals that we call the collective contact intervals that are a fraction of the whisk, where we think the rat is actually actively acquiring data. We call this idea windowed sampling, right? That there's a region of time that the whiskers are actively acquiring data. And so some advantages of this windowed sampling idea, that it's taking data only during this very windowed region, is that it may be integrating spatial information about the object within a single temporal window. And that eliminates the variability in how the whiskers scatter onto the object. And that occurs because of the changes in whisker velocity. And what this would allow the rat to do is to gain an initial impression of the object with a single haptic glance, regardless of the precise temporal structure with which the vibrissae happen to make contact with the object. And then you don't need memory to perform temporal integration. And if the rat's perception of object contours doesn't depend on temporal structure of sensor contacts, then the variations that do occur at that time scale could be used to detect object motion or texture. And finally, it resembles saccades, right? It resembles this periodic sampling strategy that's similar for saccades, and also sniffing behavior. So maybe you're familiar with Zach Maynan's work, looking at the sniff as a unit of olfactory processing. We think that the same thing is happening here with the whisk. And the separation of encoding and retrieval of memories in the hippocampal system. So we think that this behavior is representing as potentially a correlate of the neural oscillations that are happening in cortex. So, and I realize I really pushed through that last part of the talk a great deal. I skipped over a lot of the details. Happy to go into any of them with any of you. But that's sort of the place I'd like to end here, is that these are the neurobiological ideas we're pushing forward with this behavior. So thank you.
Yeah, I was going to ask you about the multiple effect before you.
Sorry, the what?
I was going to ask you about the multiple whisker effect before you talked about it. I mean, it seems to me, the Kinect works by effectively projecting an array of lights and sensing the deformation to get depth structure. The Microsoft Kinect, the game thing. It's this new game thing for the extras. So basically, you can wave your hand at it, and it senses your depth, and it does it with infrared light. And it basically it projects a circle, and depending on the deformation of that, it can tell the depth structure of that object. And it's got an array of them, and it just seems to me that some of the stuff you've got this visual array. It seems a bit more visual to me than, I know it's coming through haptic stuff, but it really seems like it's getting a picture. I just wondered how you were going to integrate. You've got this beautiful arrangement, and I'm sort of going to do some sort of, I don't know, can you reconstruct sort of this thing from multiple touches? Is that where you're going?
Yes, we can,
Okay.
is the short answer. Yes, we can certainly, engineering-wise, we can reconstruct a 3D object, period. I'm confident we can. We've done it in simulation. We can do it in hardware. Right?
Yeah.
Now, whether the rat uses the exact same mechanisms to do it is an open question. And we're recording from different structures that I hope will tell us. Lots of people record from barrel cortex. And I really like the idea of figuring out the input before going any higher. And when I started the lab, I felt like, look, we don't even know what information these whiskers are getting. How on earth are we supposed to figure out what cortex is doing if we don't know the input? And if we can characterize the neural activity in the ganglion, then at least we have an idea of what the sensory variables are that are important to the animal. So we're headed there first. One idea that we had was, look, record from the ganglion, let the rat explore an object, reconstruct the object based on the signals that you're getting from the ganglion, right? Obviously, we can't get enough signals to be able to do that, really. We'd have to do use them as some sort of basis set and do Monte Carlo simulations pretending that we had 50 or 100,000 neurons to reconstruct that object.
And you also, you said initially about the stereo sniffing, which I think is fascinating.
Yeah.
And do you think they're doing kind of stereo?
Oh, great question. One of the propensities we found is that the rats tend to explore the object more with their right whiskers, and that's consistent with a paper from 1994 by La Mandola and Bever, who showed that rats learn better with the whiskers on their right side. So there are certainly cerebral asymmetries in the rat. There's a paper from Nicolailis's lab looking at cross-cortical, cross S1 projections, bimodal, bihemispheric symmetries in the rat. It's a great question how it integrates information across the sides.
I got one last question. So this is kind of related to, I mean, Tony and you, I don't know, I'm getting the impression of slightly different ways of using the whiskers, some more active or something. And I just wondered, I may have missed it, but what muscle control have they got? Can they move them sort of each independently in 3 dimensions? I mean, what's the control that they've got of them?
So I'll take a stab on that, and then I'll let Tony give his impression too. So you can move your thumb independent of the fingers on your hand, and you can mostly move your first finger independent of the other fingers on your hand. But when you start to move your 4th finger, it's you can't move it all the way down. It's connected to your 5th finger, no matter how hard you try. So similarly in the rat whisker system, rats can leave their caudal-most whiskers completely still and move just their rostral whiskers. This has been shown by many labs, so that's known definitely. Leave their caudal whiskers still, move their rostral whiskers. Alternatively, they can leave their rostral whiskers still and move their caudal whiskers. So it's like the thumb and the pinky. I can move completely independent from each other. But when you start talking about whiskers in the middle of the array, or whiskers that are neighboring, then it becomes trickier. Then it's not clear whether the whiskers are completely independent or whether they're connected. There's work from David Gollum's laboratory in Israel who showed that the minimum unit that can be moved is 2 whiskers. And but you can sometimes get 2 adjacent whiskers that are going in opposite directions based on the same muscle contraction pattern. Then there's data from Ahisar's laboratory showing the whisker roll. The whiskers can roll like this. And that roll, they locked to the theta rhythm, to the theta whisking cycle. So the roll is not, according to the Ahisar laboratory, the roll that the whiskers do is not independent of the protraction. So it's clear that they don't have the full dimensions that they might have. They can't roll and protract their whiskers independently, at least according to the Ahisar laboratory. And work from Ziegler's laboratory has shown that the dorsal ventral component of motion is very tiny compared to the rostral caudal component. So it's primarily one-dimensional with a little bit of roll. And Tony, did you have anything to add to that?
Yeah, I think there's intrinsic muscle for every whisker, but the muscle actually connects 2 whiskers within each row, within the row, so it connects across the pair, and that's why if you move one whisker, you can see an effect on the neighboring whisker. The facial nucleus, which has the motor neurons, it's not clear whether there is the structure within there for individual whiskers to be controlled. That I think still has to be more fully evaluated. So certainly there's a lot of coupling. And then for the retraction, that's largely the responsibility of extrinsic muscles which move the whole pair together. So there is the capacity for some differences in the movement of single whiskers, but in general they're moving more or less together. But one of the things we see is that the velocity, particularly of the more caudal whiskers, it's usually slower than the more rostral whiskers. So as the whiskers move forward, they spread out because the back ones are moving less, and that's partly mechanical. But sometimes they seem to be able to move their more caudal whiskers more quickly so that they move together. So that the back gap closes between whiskers as they come forward. So there's a lot of degrees of freedom of control, but there's also a great deal of coupling.
So this is what Tony was talking about with each follicle whisker complex has an individual sling muscle that goes around it, but they're connected up to each other. So they're independent but not independent. This is from 1984 and it's in the mouse, but we think similar. They're seeing it in marsupials as well.
In the work you were describing at the end, there seem to be 2 different aspects of active touch. Well, at least 2. One being that all the whiskers were controlled to contact concurrently, and another being the sort of head movement, overall head movement, the saccade.
Yeah.
Would you?
They're sort of
agree with that?
happening at different scales, right? So the head movements are happening at the scale of the head, right? So
Yeah.
make sort of big leaps like this. And the whiskers collectively coming onto the object are happening at the time scale of a single whisk.
And you see those both as active touch?
I'd see the whole process as active touch with the rat
okay.
exploring this object.
So Mitra, the paintbrush
Yeah.
uses, does it do sort of continuous sampling of the surface?
Yes. Yeah.
Okay, so but your hypothesis about the rats is doing this more like snapshot sampling.
Yeah.
So if you can do continuous sampling with the paintbrush, why not?
Why not? Why can't the rat do it? Why can't the rat do it? It could. It's just, you know, our data doesn't. We need neural recordings
Yeah.
to say you can read it.
And as you said, it can then do texture as well, can't it?
Sorry?
You can then do texture as well with the small scale tempo.
Yeah. If you're making use of the high frequency stuff to do texture or motion. Yeah, right.
So, a number of years ago, there was a series of studies about the cricket sourcil system, where they were using fine hairs to be able to discriminate a lot of information about the nature of airflow.
Yeah.
And one of the things that really struck me about that research was that they got a rich amount of information by looking at the different channels that reflected the material stiffness of the hairs. And so they could really reconstruct a lot about the characteristics of the airflow of that. Is there a role for the stiffness of the hairs in the amount of the kind of information that the rat can pull up?
Well, the stiffness is the product of Young's modulus and the area moment of inertia. And so the stiffness of the whisker, even if Young's modulus were constant along the length of the whisker, the material were constant along the whisker, which it is approximately, not exactly, then the stiffness would vary based primarily on the taper.
I was speaking specifically about different hairs having different stiffnesses.
Different hairs have different stiffnesses because they have different diameters. Yes, that's true.
And is that a potential source of information?
Yes, because that's. See this quantity C in there? That C is a stiffness-related constant. And so you're going to have different whiskers that are sensitive to different different DMD theta based on that C value, which is directly related to primarily to the diameter of the whisker.
And it also affects the resonance of the whisker as well, doesn't it?
The diameter and the length will also affect the resonance.
Which people have said can be used for
And-
sensing.
and-
Has anybody looked at that?
For flow?
I was just thinking, in general, even the contact might.
Yeah. Quite a lot.
Yeah.
When you talked, you mentioned it, I thought it was an obvious thing that.
Yeah, no, people have definitely looked at the resonance of the whisker, and when you blow air over the whiskers, over rat whiskers, not over seal whiskers, the dominant effect you see is the whisker resonance. There's conflicting data on whether the resonance of the whisker is important during texture discrimination. And one of the properties we think is extremely important that has yet to be quantified is the damping of the whisker. We think we have evidence for structural damping, which is an amplitude-dependent damping, different than viscous damping, which is velocity-dependent, as you know. And people don't know really how to deal with structural damping very well. We're just headed in there. Maybe you know structural damping and beam mechanics. But you probably deal with damping that's proportional to velocities, right? You have viscosities that are proportional to velocities.
I haven't looked at very much at structural damping in, for example, superinteraction. So I can give you an example.
There's structural damping is big in airplane wings, which is why I thought you might know of it. It's a complicated thing that we're struggling with right now. Damping is hard.
Any last question? Okay, well, I would like to thank both of our speakers again.